Important lecture today. We'll have two guest speakers. And the idea is that... Hey, what's happening? Very nice. This one doesn't want to... So, it's correct here. It's not correct there. Anyway, the most important names you see. That's Doutzee and Erik van Dijk. Erik, make yourself known. There's a finger there in the back. You see that? That's Erik. In the back row there. You'll see him the second hour becoming active together with Doud. But he had meetings, so he'll come really for the second hour. So I will fill you in on the very brief history of genome sequencing and the Human Genome Project. Then I'll sketch the problem of sequencing, genome assembly, how to get a true copy of DNA from how you measure all of the data. There are two basic forms to do that, and I'll zoom in because that is important for your second practical starting today. A method to do reference sequencing and a special way of indexing to make it scalable. So, what you really are going to do with it, you'll study for your second assignment, you'll study copy number variation. From now on, CNV, that's what it means for you, okay? And you do this based on a method that has been developed at the VMC. And the two gentlemen in the second hour will explain all about that. And that method is called qDNA-seq. And I hope I'll get the top part on it tonight because it's here, but not there. Okay, so the bioinformatics Big Bang, what was it? It started in 1990. There was a press conference in 2000, so 10 years later something had happened. Who has an idea? This is a Big Bang, right? I'm impressed by my fantastic animation. Anyway, the Human Genome Project. And here you can see, if you look at the blackboard there, that, when they started this. So in 1990, people said, it would be nice to be able to get the genome of species, because there is some information on DNA. And then people said, that's crazy, we can do 3 base pairs per day in a big group, so if you want to scale up to 3 billion base pairs or something for the human genome, utterly impossible, will not happen in a million years. So by 95, they had a couple of genomes, techniques had sped up, and it seemed, still it was considered crazy, but some people were crazy enough to be daring enough to say, look, something like this, I think something can be, we know we want a bridge, we don't know quite how to build them, but a miracle occurs, but then, and this project has been astounding in the sense that it has become, in the end, much cheaper than planned, and it was finished sooner than planned. I don't know if you've ever had a bathroom constructed in your home, or your parents, This never happens, right, for any normal project. It's always more expensive. And twice as long or something. So in the end, of course, certainly seen for health, human health, the human genome has been important. Maybe I've shown you this slide, but I like this pop art type little picture here. Okay, so what had happened? Let's go quickly through the history. Here you see this important date. I think it will come up 26th June in the year 2000. The press conference led by the then US President Bill Clinton. In the back there you see this dark face. Sometimes I call this man Dark Vader. There's a special story about this gentleman there. The name is Craig Venter, and by the way, incidentally, you see it was 2000. Do you see that type monitor there? Weighs a ton. It was called television, I think. Old days, right? Old school. Anyway, you see there the then Prime Minister of the UK, Tony Blair. A bit in the background, but he wanted to be represented as well, of course, as politicians tend to, wanting to do that. What had happened? This was a great idea. Let's work together and let's create this milestone for humanity that was expressed at this press conference. So let's start making the human genome happen. Now, what had happened? In NIH, the National Institute for Health in America, there was the gentleman by the name of Craig Venter, you see him here, and he went to his then director and he said, I don't know if it has occurred to you already, but I'm a genius. And my colleagues all recognize that. And I think a genius deserves to get a big team because I want to pull off this sequencing project. I have ideas how to do that. Give me 20 staff positions and I'll punch an army of postdocs and so on, and I'll pull it up for you, and you will shine, and you'll be thankful to me for the rest of your life. So this director said, Craig, yes, I'm sorry, of course you're a genius, but there are more people here, and maybe somebody else wants something, so what about 2 positions? So that was outrageous in the book of Craig Venter. So he said, look, I'd quit. You'll regret it ever afterwards, but I'll quit. You know what? I'll found a company, and I'll do this, and now something else. I'll patent each and every gene that I will discover, so that you pay dearly for any future research in this arena. So here you go. Craig Venter's threat. Made proprietary my patents, cellular genomics, and everybody thought this is crazy, of course. And also, how can it be that that you would sequence a human genome? How can I sequence your genome? That sounds outrageous. So let's look at the legislation, and you find out that you cannot just patent somebody else's personal information. It seems like an easy thing to understand. So they checked, and of course there was no legislation. There was nothing. So what do? Trust that it will happen, that it will come and turn to the good just on its own. And then people said, oops, Venter had gone underway already, and he had already sequenced snippets of information, and it looked good. They thought he was, he fantasized a lot, but it seemed real, so he was really an opponent from the allies, the universities and research institutions. So he was an enemy to reckon with. I mean, an enemy, a competitor, let's put it like that. So what happened? It was in the end. I'll give you one little other thing. I need to look. Eric Lander. So Eric Lander told me the story. Once he was phoned on a Thursday morning by 9 o'clock, and he answered. He takes up the phone, and normally you say in America, hello, you say full name here, but then you say, and then the other side of the line said, hello, yeah, Mr. Lander, yes, it's Bill Clinton here, Mr. President, what can I do for you? I hear there's a problem. Okay, yes, true. You're in a race. I want you to win that race. What about $5 million? Could you use that? Yeah, certainly. So there you go. So politicians handing out, and Clinton single-handedly handing out chunks of $5 million. There was nothing like it. And he had given out those chunks to a couple of people. I think he handed out 20 million in 50 minutes or something. Well, for Americans, but still, this is money. And so that was astonishing. Politicians seeing the urgency of something scientific. That doesn't happen very often, but here that was the case. So in the end, they were racing. And it was an unfair race in the sense that there was a lot of research institutions against a single company, but that single company had attracted a lot of venture capital. They had bought up the best computer scientists in the country, and they bought the largest cluster computer ever installed they had. So, they're doing something. It was unfair in another way that the allies, the Human Genome Consortium, they were called, they put everything up on the internet. In a way, you could say the project could only succeed because in the meantime we had internet, we had IT technology, right? Very, very important. But of course Venter looked at all of this data, checked it and tried himself, and of course he didn't open up on anything because it was all, kept hidden. So anyway, so what happened? There was a race. These are 2 papers where you can see after, in 2001, after the dust had gone down a little bit, where you see that the Human Consortium published in the, one of the most important journals, Nature, in the UK, and this company, Craig Venter, published in another journal. So this was the same week, so there was a bit of talking beforehand, of course, to make that happen. So what had happened, and let's show you this, the press conference, show you pictures in a moment, after this bitter fight, and it seemed to go completely wrong. Craig Venter was winning the battle. And I'll show you a slide in a moment. This is during the press conference where Clinton again, he saw the importance of this. These are beautiful words that president could speak. You see that? And so very nice. Here you see the press conference. See all televisions here. Here is Clinton, and you can't see this really. This is Craig Venter, the private person, the cellular genomics. Here is the director of the NIH, the director of the Human Genome Consortium, and Clinton was a fairly strong man, and they seemed all friends, but you had to keep them apart. They wanted to kill one another, at least attack one another. So here you see Tony Blair once again a bit in the background, and here you see this is a remarkable moment. Those two guys shaking hands. I think, you know, considerable tension there, as we tend to do nowadays, right? Good practice. Anyway, and here you see that again. Okay, so let's say all is well that ends well. But I'll show you the drama. We're all jolly friends now, of course. But Jim Kent, they were in despair. The Allies were in despair. It was in already June, and they didn't have genome. They couldn't assemble it. They lacked the software. Where already Celery could do it, they couldn't. So they thought. So there was a smart guy sitting in his, I think, in his loft, somewhere in Santa Cruz, building the software. And here, the press conference was on June 26. This assembly was done on June 22nd. 4 days before the press conference, they pulled it off. We won, at least we can do it. So here you see, this is Jim Kent. Leave the guy alone, he writes software, he doesn't want to talk, but he's great. So he could be called the savior of the Human Genome Project. He wrote a program called Gig Assembler. And here you see some important dates. June 22, he pulled it off. He built the first assembly of the genome. Celery, they had their complete copy, they had all kinds of versions, but 3 days later, on June 25, and they started discussing, and the next day, there was a global press conference. Can you see? This is, this is people moving, and Clinton saying, I want this done, right? And then it happened. So here you go. So this was really hair-raising, what all happened. And so, we don't need to patent, because they call this a photo finish. And they did it all together. That's now officially in the books, of course, and so on. Okay? So, the Human Genome Project. What did they do? What were the differences? So, Craig Venter was a Vietnam veteran, and he called his technique the shotgun technique. You know, he was used to that kind of stuff. What was the idea? And this was considered completely crazy when he started it. You take a big DNA, a complete chromosome, millions of letters in a string. You cut it you shoot it to pieces, shotgun it to pieces, with sonification, all kinds of things that make DNA break and fall apart in small snippets. And then it is this. So these are all short stretches of DNA. It's now all single things. You don't know what the order should be. You have snippets, right? And that's basically the puzzle. How can you get from here to a sequence that is just sequential in line, where all of these snippets should somewhere land here? Yeah? And here you see already an idea as to how that could be done. So this is what Venter suggested and carried out in the end. Everything shot to pieces, and then you assemble it using large computers and smart programs, and then you've got something. Now, the Human Genome Consortium, all of the unified institutions, did it a bit more, careful. They used back clones, artificial clones, so they built snippets of DNA into a bacterial genome. What comes from that they had chunks of 150 kb, short, longer chunks, that they sonificated, they shot in pieces, but at least they know that all of these are in this stretch of DNA. Here, you don't know where this is at the total beginning of the DNA, total end, you have no idea. Here, at least you know it's in that chunk of 150, and this bunch here is in this chunk of 150 kb. They know the order, so their problem was less. Their puzzle was a bit smaller, right? And so this is more careful, more frightened, you might say, not as long as here. But this was considered completely unfeasible. In the end, they did it. They made the technology. The basic technique to sequence each of those snippets was done is, of course, molecular biology techniques and so on. And that was Mr. Zanger, who got 2 Nobel Prizes in the same field for chemistry. Quite amazing for sequencing and for solving insulin protein. He was maybe really one of the first bioinformaticians, really. There's an important institution called after him, the Zanger Center, close to Cambridge in the UK. So he had this technology. With this technology, you can sequence snippets of roughly up to 1,000 nucleotides, 900 to 1,000. So you have snippets like that, and then you need to build those together. Okay, so here again in words what I just told you. Just a little picture, this is DNA, right? chromosomes, so you got the feel. Here is sort of an outcome of the Human Genome Project. There are, of course, humans have 23 chromosomes, one of which is the sex chromosome. There are roughly overall 20,000 genes. Still a debate how many exactly, meaning we still don't know quite the picture in precise detail. And so there were 90%, 2.9 out of 3.3 billion base pairs were sequenced by the end of the Human Genome Project. Not everything. Okay, this is sort of interesting to see what kind of DNA things there are. And if you look at the green bits, it's more than half. And these are the bits of DNA that are repetitious. I think I told you that we are big stutterers when it comes to our DNA. A lot of fragments repeat themselves, and it's more than half. Okay? Get back to that, because that is a problem for sequencing. I'll show that later on. So, anyway, nowadays we got new technology since then. The last few years have seen tremendous developments in technology to sequence. It's much cheaper. The first human genome cost roughly $3 billion American dollars, and now we can do it for $80, right? Sequencing human. Maybe 100, it will come down, it might become 40 in not too long. So it is, and it's much, much faster. Where the Human Genome Project was supposed to last 10 years, it turned out to last only 5 years, twice as fast as planned. Now you can do human genome in a matter of a few hours. Not bad, right? It is cost. So one problem though is that compared to the human genome where snippets, fragments, reads as they're called, were roughly 1,000 long, the reads here are a lot shorter, 100 to 150, with the most dominant technology. Smaller bits. So the puzzle problem is bigger, so we need even bigger computers. Okay, here you see a nice slide. This is here, the speed is here. And here you see the length of each of the reads that each of these machines or technologies produce. Here is Zawer. I told you roughly 1,000 for the read length, but you see it's very slow. This is logarithmic, by the way, so this is really orders of magnitude faster. And the most widely used technology is called HiSeq. Here. And technology, and I think, Eric, is that what you take your measurements from? HiSeq? Yeah, there you are, this one, okay? Here is interesting, you see that we get now technology for larger reads, and of course, if you will be able to just take a chromosome and sequence it in one go, we don't have an assembly problem anymore, right? If you can just measure the whole thing, but not just there, but you see already that the read length is approaching 10,000. And the next generation technology is underway where this even will be longer. Now, to show you, I don't know if you've seen this, this is the Lumina HiSeq machine that is used for the data set that you will use for your second practical, your second assignment. This is a big screen, this is roughly as tall as a human. This is a new sequencer, Minion, nanocore. This is as large as a memory stick. You can take this in your pocket. Take a little suitcase, I have to be honest, with a bit of molecular stuff, and then you can go to Africa and sequence the Ebola virus. So you can really go on site. So technology is becoming tremendous. Very important to do that you can have moving labs, because infections and those kind of things, they don't wait until they're in a laboratory. You have to go into the fields to measure. So very important, this technology. They're doing well, these people. It's a new model for industry. Anyway, we're bioinformaticians here, at least for today. This is the main problem. You have lots of snippets. This is a single DNA, just cutting little pieces. And now the idea is, in the end, this came out of a DNA sequence that looks something like that. So how can we reconstruct it? How do we do this? Anybody an idea? Tell you a quick story. 2 quick stories. Version number 1. You're up in the loft at your grandparents. You find a very old book, you think. You go to a connoisseur. This gentleman tells you, you know what you have here? I don't know. I can't read the letters. It's very strange, everything. Well, it's a copy of the Gilgamesh epos from the old, from ancient cultures. You want yourself $1 million at least if you sell this. You think, oh, that's good. So you find somebody wants to buy the book, and the next morning this person comes to collect the book, and you will get €1 million. Feels good. So you're sleeping nicely during the night, not knowing that there was a big mouse coming into the house. He likes old paper. started eating. All the pages were gone, and the next morning you find a pile with, it didn't like ink, the mouse, so it respected the letters, but, it was a pile of junk. And how can you read? You don't know the language. Can you now read the Gilgamesh Epos? Difficult, right? Can you ever reconstruct the book? Tell me. Say no. Be courageous. No way, it's a pile. You don't know the language. What is after what? Should be a single sequence. No chance. Okay, story number 2. Even better story. You're up in that loft again. You find 10 copies of that book. Exactly the same. That's what you can see. So this tells you, now you earn yourself 10 million. That's, even better. So you sleep even better, not knowing that an even bigger mouse was there during the night, eating the 10 books. The pile in the morning was even bigger. And now what? Small chunks of paper. Is there a chance? What's your answer now?
Maybe.
Yes! Say that. Why is that?
You could leave them together.
What do you use?
Reference.
Put them together. I like what you say. Well, let's see if you agree with this. So, anyway. Okay, what we're talking about is called de novo assembly of a genome, right? You have no reference, you know nothing, you need to work with the snippets of paper in this that you have. Okay? So let's do that first. What about this? Does this come into play, you think? You don't know language, but you see maybe, hey, this snippet has ends, or this snippet from another book. Let's say the mouse is eating randomly, right? It didn't eat each of the books in exactly the same way. It was hopping away in another one. So this might happen. Now if you find this, then you got something. Maybe you can fit all of these pieces, cut them into one, glue them into one another, and in the end, you might have reconstructed the book. I don't think you get 10 million anymore, but maybe you get something for the message. Okay, so here you go. If you do this, then you get this sort of stuff, overlap after overlap, and perhaps, if you're lucky, you can build a longer string like this. Okay? That's what can happen using overlap for de novo sequencing. But of course, there are problems. Maybe the mouse in the end has been eating away larger chunks of the complete book. It's one possibility. I'll show you a picture. And the other one is the one I already referred to, the repeats. I'll show you why that is a problem. Here you go. This is what can happen if you get, that's what people call lack of coverage. And this is, sometimes DNA is resilient against sequencing. There are parts of DNA at the centromeres between chromosomes where it doesn't want to work really. So there are often positions in the DNA where you don't get any information, no reads. What will happen if you do this? If you assemble each of those, if you get these overlaps, you call that a contig. Comes from the English word, of course, contiguous. All these fragments, you get an island, right? Where you know, okay, this is a chunk of DNA. I think I've reconstructed it. So here you will get 2 of those contigs, because here you can't do anything. There's no overlap. And then there is another problem. This is called contig 1, and this is called contig 1. 2, but in reality, you have no idea which one is first and which one is second. You don't know. Okay, keep that in the back of your mind for a moment. And here you see that, so here we go again. Try to follow the message once again. One contig, another contig. And if you don't have information in between, you get a gap. And what you see here, of course, when you do the exercise, you only have this stuff. This is your data, right? In the end, you would like to reconstruct, of course, the complete DNA, but you don't have it at that moment. That's what you're trying to get. We map it here to show you the problem. This is a gap. But in reality, you don't know how large the gap is and even the order of the contigs. Okay? Would like to do better than that, right? How can we solve or do something about that problem? Alleviate it at least. Okay, so how to deal with gaps. And so overlapping unconnected contigs, yeah. And now, already Craig Venter, you mean Darth Vader? He already started using something called paired end reads. And that's a fairly clever little thing that comes to the rescue here for a while, to some extent. So what is a paired end read? Here you go. This is the idea. You have a very long stretch of DNA, maybe 10,000 long. No way with the old technology you can sequence it. But then people reasoned, you know what? Maybe I'll sequence the first 400 from the one side. Maybe the last 400 from that fragment. We do this by the complementary strand, but anyway, you can do that. And the nice thing is that you can measure, you might say, resolution is good enough in technology that you can get the exact number of bases of a fragment. So what have you got? Maybe this fragment is 10,000 long. This is 400 and that is 400. So what is exactly in between? 90, 200 nucleotides. You know that. So you know the length of the intervening region exactly. Okay? And this you could map, because here you have the letters. Here you don't have the letters, but you know how many. And here you've got the exact letters of the red bits. That's called a paired end read. Yeah? The ends are paired, you know. So, sorry, I go the wrong direction. There we go. Yeah, so there are two kinds of paired end reads. They're called paired ends, so, and made pair doesn't matter so much, but so you know, paired ends are a bit shorter than made pairs. It's the same principle, but here you get it. You want it, it's for reference. I'm not going to linger long on this one. Here you do something more difficult, molecular biology-wise. You circularize the genome, then you can measure. So here there could be 2 to 10,000 bases in between, and here these are a bit shorter in between. So what could you use? So how can you already get an inkling how you could use them? Suppose you have two contexts where this bit you know is sort of, you can find here, and this bit you can find in the other context. That gives you some information, right? I'll give you another picture. Here you go. If these are the paired end reads like this one, and pairs with that one, and the intervening stretch is 8,000 or something, right? It might be that if this bit lands in this context, and the end of that paired end read, the other paired end, and lands here, you say, hey, that means no, the contexts are like this and this. This one is preceding that one. And then you have maybe another context, and you could do the same trick with other paired end reads. And the more paired end reads you have, obviously, that link this one to that one in a consistent way, with the spacing okay, that gives you extra evidence. Right? Paired end reads really help to locate, to order these contexts with gaps in between. Not only the order, but even if you with a bit of luck, you can estimate the spacing in between using paired end reads. Because you know, once again, you know the exact length of the intervening fragment. Yeah? Okay, so this is one. So, here another thing, what happens? What can happen? Here is a genome, and in reality, it's a stop-ring genome. Here is a fragment, and that fragment here is exactly repeated. What happens if you just get all these short snippets, and you don't know that this is the case in the DNA sequence? What will you tend to do? This. You will think, this is a fragment, here's another fragment, all these reads, they are consistent with this, very good. These are just copies of the Gilgamesh epos, you know. The different minds, so it should be like this. Well, in reality, you not knowing, it should be like this. And what then happens is that if you find this in the end, if you do this, then obviously this bit of DNA you don't know how to handle, that is normally thrown out. You're left over. Does it work?
The battery is almost empty.
Was totally charged, it was sad, but well, let's see if it stops, let me know, then recharge it. Shall we try that? Maybe it holds out for the first hour.
Oh, thanks.
Ooh. Fantastic. I'll talk on and you try and do this. So here's another way to show this. So if you have these two repeats, and this one should have been measured here, but you don't know that, then you tend to build them together, you lose the second repeat. Whereas if you have a so-called paired end read, and this green one lands here as it does here, then you know, if that red one is here, then something here should happen. Paired end reads really help. And so that is what Cellular Company mainly used in their sequencing efforts, and it's really a very good idea. So this is used a lot nowadays, paired end reads. Okay, this is just, most of you might know this by now, but the more copies of the Gilgamesh epos you would have, obviously, the more certain you can be that your reconstruction, I'll get to you after my sentence, so the quality will be better. Yeah, of that. So that's called either depth or coverage. The more expenses you take, the more reads you have, the more evidence you have to reconstruct your DNA faithfully. Yeah.
How do you know the length of the part of DNA that you were missing?
With the paired end read.
Yeah. Like
So,
previously?
yeah. This one, you mean?
The one before?
You mean this stuff, right?
Yeah.
Yeah. So you have a fragment of 10,000 long. You know that. You can measure that. You don't know the letters. It's a fragment. But you can weigh it or do something that you know it's 10,000 bases. Now you sequence the first few hundred and the last few hundred. Then you know exactly how many bases there are in between.
So even if you cannot sequence
Yeah, you don't know the sequence of this bit, of the black bit, but you know it's 8,000, right? And you have the sequence of the red bit. And that red bit could land in one contig, and this red bit could land in another contig, and then you know how they are held together. And you know the order, and even the spacing. Yeah? It's important that is understood. Okay? So if you don't know how to do it, you just say we need an algorithm. Talk to an IT guy. He will help us. So, here is your problem. Millions of snippets called reads. Process, okay, something you think about to do it, and then you hope in the end that you get the alignment of all those reads, and of course the DNA sequence of the new strain, because this is de novo assembly. De novo means you're sequencing a DNA of a new species, you have no clue how the DNA looks. Another problem is if you are sequencing another genome of a human nowadays, we have millions of the sequences, so you can compare with what is there. That's a different problem. Okay? Now, here you go. What will be used is graph theory. Who has here seen all these pictures? How many? Not so many. Yeah, a few in the record. I'll be quick, but I'll try and make it clear to you. Okay, so the Brown graphs. I'll skip over this. It came from Euler, a mathematician who had a problem that he said he lived in a town with a lot of bridges, and he wanted to do a walk every day, and he said, I would like to be able to walk over each bridge once and get back from where I start. Is that possible in Königsberg? And if you look at this, you can quickly check. The answer, by the way, is no. But he needed to invent graph theory and this way of representing a problem with nodes and connections, edges, to do it. So, for this problem, read this and read the paper if you want. So this all graph theory comes from this, and algorithms to do the Eulerian path. Can he walk over each bridge and so on and get back? You make an Eulerian circle. That is what it was after, and so on. So we're going to use this stuff to find overlap between each of these snippets and see how we can reconstruct a complete genome out of millions of fragments. Now, how do we do this? In a fairly simple way. If you have a stretch of 4, so we break up each snippet, be it 150 long or 1,000 long, depending on the sequencing technology used. You're going to break it up in small fragments of, say, words of 3 long. They're called k's, and k here is 3. And if you have a word of 4, ATG and TGT, you can easily see that the first 3 letters are ATG, and the second 3 letters are TGT. So what you can do is break them up in 3 and make an arrow, meaning this one follows that one, and the overlap between these 2 words, as you can see, is 2. The last 2 letters here are the same as the first 2 letters there. And the complete word reconstructed by these 2 k-mirrors is ATGT. Okay? That's the. So we do that. And here's a bit of definitions you can just read. So if you have a snippet like this and you do this, you can already see you start building a graph with nodes. Nodes are these snippets, these k words, 3mers, words of length 3. C-A-A-T. Who can tell me what is on here? C-A-A-T. Very good. Okay? And so on and so forth. And this means for real case of 1,000 this becomes larger, but this is just. So now what? If we add this one, what will happen? We'll see something. You see overlap?
Yeah.
So in red is what is now double. ATGGC. ATGGC is here as well. So this bit should be cemented on, right? These 2 are the new blacks. Yeah? So, and what have you got? In fact, 2 overlapping reads, and this is what you want to do, right? So this is how it starts working. Now, then there are algorithms, and this is your paper exercise. We have time, want to do this for de novo. So if you would build these snippets, you go like this, do these steps, and it's and you get this, you get all kinds of things. So, I'll show you the answer in a moment of this. So, but now you could have something like this. What do you think this might mean? What is the problem if you measure stuff? You might get errors. What if in a snippet, in a read, there are errors? What will happen? You'll be less able to match them, right? It's not the same anymore. So do algorithms need to be able to keep track of that? Well, probably. Do that. Very often they don't. So here is an exercise you could do, called the home exercise. And here is one of the answers. And this leads to a very nice graph, a circular graph, if you try and solve this. Here's your answer. And this is, when you have this fragment added, what do you get? You get a nice sort of train right here, piece of graph, a circle, and then these letters. And you might say, what should it have been in order to be totally consistent? You can see, guess what the A should have been. If it was a C, life would have been different. It would have just been part of this here, and now it's like that. And this is what you do, what you get if you use this technology. And if I blow that up, then you get sort of pictures like this. And in the end, if you want to reconstruct, you should choose one of those. That should be most supported by the data. Okay? And this is even larger than this stuff. And that's sort of, all kinds of nice words, tips and bubbles, tip and a bubble here. And this is if you do two big genomes, you align them, you get this stuff. Yeah? It's all by that algorithm, which is invented by a Dutch guy called De Bruijn. De Bruijn graphs. Okay, so now the second step, and this is more important for your second practical. I'll speed up a bit. This is if a reference genome is available, so you have a genome of a human, and you measure another genome of another human. Maybe the human is quite like the earlier one, maybe not so much. But on average, the difference between two humans, I think I told you, 1 in 1,000 letters, right? Roughly. Okay? So it's not dramatic. That means comparing it might help you place your reads. So what's the game here? It's different. You can read those papers if you want. And here you get an inkling of what is happening. These are reads. Here is your reference genome. You've really got that genome of another patient or another individual. And now you can, instead of trying to find overlap, you can just say, no, I just align each of those snippets to my reference genome and then see where it lands. So that means the order is, in fact, clear from the outset. Because, that this one lands here, this is the next one landing a bit further downstream, and so on and so forth. And you can also see the depth of your coverage, how many pieces of evidence you get per position, and that is here, I think, indicated here for this, that this is a region that is well sequenced, and here a bit less, and so on, and here nothing, right? And here you see this blown up, locally, alignment onto a reference genome, right? Do you know how to align pairwise snippets? What's your algorithm? Dynamic programming, finished. You think, do that? Align millions of reads onto a genome. How long do you need to wait? Well, more than a lifetime to get it done. That's not enough to cure a patient, so that's not a good idea. So we need to speed up, right? So there is technology to speed up. Okay, your input files, by the way, are quite large. They're called BAM files. Reads that come off sequencers are called BAM files. There's an input file you use. So basically, for your practical, you'll get measured reads. And you have an assembly problem, and then you get biology, meaning about copy number variation, and the second lecture will be about that, second hour. Yeah? So, technology here is called Burroughs-Wheeler transform, and I'll quickly try and fill you in on it. So, I already started alluding to that, the problem of mismatches. A reference genome might contain mismatches. The two individuals might not be the same. 1 in 1,000 is different anyway. So you will have reads that do not align perfectly to a genome. With dynamic programming, is that a problem? Can you deal with mismatches and gaps and so on? Easily. If you really want to make quick technology, maybe that's a bit different. Okay, so you cannot do exact string matching in biology because you won't be able to match. Yeah? So the technology to do this is called Burroughs-Wheeler aligner. And it's in fact a clever sort of indexing technique, and I'll try and make that clear in the last couple of minutes what it really is. So, this is a phrase from the paper, fast and accurate short read alignment with Burroughs-Wheeler transform. It's a very fast aligner, as I said, it's based on indexing, and it uses something called suffix trees. And I'll show you that in a moment. It can handle inexact repeats. I'll show you an example, and it's all described in this paper where everything is explained. And here you see, I told you about citations. It's not the top scorer. They were at 50, 60,000, but 16,000 after a couple of years is not bad. Okay. It's heavily used, as you can see. All right, here we go. The trick is, for now, this is our genome. The genome has the letters Google, right? Just don't know why these guys came up with this. So now you do a trick. You order them circularly. You say Google, and now you say I put a G to the end. What do you get? O-O-G-O-L-G. And you do it once again. You make all circular permutations. Yeah? And then you order those alphabetically. So if you look, you see Google, where's the second one? There's this one, where's the third one? Probably this one. Where's the fourth one? Starting with the G, this one. Oh, here they are unordered, so this is the, okay. This is the unordered one, obviously, so here you are. And here you get if you lexicographically or alphabetically order. Yeah? You take this and then you order it, and you get this order. This, by the way, means the end. It's the end sign, and you put that up front, but then you get the two with the G's, this one and that one. So this is ordered alphabetically. Yeah? Now, and this is now called the Burroughs-Wheeler transform, and what they use to show the transform are the last symbols. You see, this is Google, this space is just to explain something, it's not really there, so these are just the last letters of each of those ordered streams. That is called the Burroughs-Wheeler transform, because that shows you basically how this is. Two possibilities according to the tree, do you agree? Where are O's in the sequence? What can be before an O? G or an O or there. Yeah.
So what do the numbers represent in these?
These numbers?
Yeah.
Yeah. These are the start. So here, I'll show you an example. Let's take this example then. What does this 1, 2 mean? That's your question, right?
Yeah, exactly.
So that is this 1, 2. What does that mean? Where could this be? 1 and 2 is here. What is 1 and 2? This one. Go or G-O-O. What can be in front of a G? Here, the start of the sequence. How can you start here, the start of the sequence, that's the start before this G, where can you be? Here or there. 1 and 2. For this situation. Start of sequence G-O. That's here. Yeah? Takes too long now to really go for 50 minutes because then we go through your break, and in the second hour I can't do that. Then we discuss over the coffee. Another question?
Yeah, so it tells us which of the options
Basically.
we choose. So is it the range or is
It's a
the
range.
option?
This is a range.
Okay.
And you see here the range is the full thing.
Yeah.
And you zoom in into the situations. Basically you get all possible substrings. So here you see the whole string here as a possibility. Google. You see that? And here you see a substring. So meaning you can immediately map reads onto this situation. It's indexed. That's the idea. And so this is what happens. So give you an example, sorry. Where was that? Here. Try and align LOL, laugh out loud, onto Google. Okay? So who can see how many mismatches do you need to allow to make this happen? Very good. Okay, let's go. Let's go down the tree. This is from the back to the start. So can we go down this route where the last letter could be a G? Is that okay? Well, one mismatch is still okay. You shouldn't stop, right? You told me. So let's try. One mismatch. What will be the second letter? Is that okay?
Yes.
Okay, so go down here then.
No.
Oh, second O. Too bad. Second mismatch. Oops, doesn't work. Go back. This didn't work, of course. This is what the route we walk. We go now down here. We go O. Can that be okay? Yeah. There's an L, there's an O, okay. And then? Oops, too bad. Go back. Go down here. Oh, too bad. Go back. And now, go down the L route. That's okay. No mismatch. The O route.
Yeah.
No mismatch. G, mismatch, but that's one. We're done. Yay! You see that? Okay, so that's what you need to do. And this is quick, but you know, still it's quite some work. Needs computation. But compared to use dynamic programming, this goes in linear time. And what was the scalability of dynamic programming? Quadratic. So this is really much better. So that's the trick to do it. Okay? So, and this is compared to a couple of methods. What is it? Oops, I should stop, otherwise we go straight into the second lecture. We shouldn't do that. Give at least 10 minutes for coffee. And so basically, but there is something useful, so you might feel, okay, where was that again? Here, look. Do you remember this algorithm? This is what you made, right? Even these guys, the die-hard computer scientists, in the end, to really map fragments that are really different, this technology is not good enough. They need to be sort of sniffed together. Yeah? Are you more happy now? Okay, that's good. Just a few questions. So what will be the cause of a mismatch? In this case, with reference genome, I can tell you at least two ways.
Error in sequencing.
Error in the sequencing, or the reference you didn't know, but you're not, your reference alignment is not a human but a buffoon. You didn't know. What will happen? Ooh, these reads, they don't match so well, right? Sometimes they try that. If you have a species that is close, you hope close enough, you could even try that. But of course, you need to realize there are places where it might be maybe too different. But it's sometimes useful. We won't do this in your second exercise right now. Okay, I think so again, we talk about really huge amounts of data. And for your second assignment, you'll run for at least 2 hours to build the assembly, right? A big computer will do that for you. Be happy you don't need to do this by hand. So error rates, lack of coverage, we know all of this. And the repeat. So the repeat problem is considered to be the largest problem. Okay? Here you got to know, is this a sequencing error or a biological variation, right? And this is take home. This is what I try to quickly run past you. Yeah? Okay, so you'll work with the BWA algorithm. You know, you can run it even without understanding each and every step. The computer knows what to do, hopefully. But I try to sort of give you some feeling and insight. So indexing, to make things fast, is all important in the whole of computer science, right? You know, what was the first index you used without knowing this? Do you remember? Let me try. Telephone book. Is that sort of known on paper? Are you old enough to know that still? Very good. How does that work? You want to find Peter Jones. What do you do? What should you know in your computer? No. Yeah, of course, but Peter Jones. Very good. Peter Jones. The alphabet. Because what do you do? P is B after. So how do you do this? You go down. Oh, too far. You go back. Oops, too far back. Yeah, that's what you do in your finding, right? It's in fact indexing. And this is just faster ways of indexing. One other thing is, I'll show you in the end. This, indexing is done all. Hey, we'll do some coffee in a moment. This is Doud, by the way, lecture at the second hour. And yeah. Okay. Hashing is one way where indexing is such that that's clever. The input is Peter Jones, and if you have that, it translates that to an address somewhere in the computer where the telephone number is found immediately. So you need to go down alphabet and back. You go there. Yeah. What happens if you have multiple Peter Joneses, you think? Oops. Anyway, you have to think a bit, but you do this over coffee. Okay? Anyway, that's it for now. We're going to listen to Doud C. We'll talk about the important biological problem, and that is copy number variation. DNA of different individuals have different structures, and that can have implications for health and so on. So it's very important you can analyze that well, and that's what you're going to do for your second assignment. So this is your induction lecture on that. So it's a good idea to pay attention to it.
As always.
Yeah, of course, but now even more especially.
Yeah. Well, thank you, for the kind introduction. Well, actually, I'm just going to do a small introduction to the problem that we have here at hand. So, just maybe 10 or 13 slides. The rest of the talk will be given by Erik van Dijk, who is seated over here, and he will take over at some point. You'll notice. So, yes, it's about copy number analysis by whole genome sequencing. Let's see. How do you feel about that? Is everybody still awake as you had quite a lecture this previous hour? Yeah, nodding heads, cool. So, what is copy number variation? So this is just the schematic of a normal genome where you can see each chromosome colored with a different color, and you can nicely see that of each chromosome there's 2, which you're supposed to have. So this is a normal situation. This is what we used to do quite a number of years ago, where we actually colored all these molecules, made a picture out of them, and actually took scissors and glue, cut them out of the paper or of the picture, and then tried to put them together and see if we actually had a pair of each chromosome. Well, quite laborious and quite Stone Age, if you would look at that now, but still it's a very nice representation of what a normal genome should look like. So enter the human genome. I think around 2000, maybe some of you were just born back then. Raise your hand. Who was already born? I hope everyone. Okay.
You're already born.
Who was beyond the age of 5? Aha. Okay. Cool. So it's actually quite some time ago already, but actually not so long ago that we were able to figure out what the content is of the complete human genome. Back then it took us like 10 years to read it out to the fullest, and it cost quite a lot of money. And what do we have? So we can print it out in a book and just put it in a bookshelf, which is not really useful. What we can do with this, of course, is a lot of cool stuff, and I think many of you are here to do just that, I guess. So let me just point out to the problem of copy number variation. So please don't mind the text, just look at the cartoons, which is of course a lot nicer. We here have a cell which is dividing itself, so it copies the DNA. And it's at the point that it's going to pull each copy of the chromosome towards the daughter cells. And here suddenly we have a carcinogen who's going to mess up the whole process. In the end, here we have the cell division, and in one cell we have too few chromosomes, and in the other cell we have too many chromosomes. And what we will then get is, of course, many of those die because we suddenly have a situation in a cell that doesn't fly, just doesn't work. We're not able to have the genes, we're not able to have the genes which we need to actually survive as a cell. So they just die. But every so now and then, we will get a aberration which does not cause the cell to die, but it actually causes the cell to proliferate uncontrollably. Or in other words, it's a tumor, it's a cancer being formed at the moment. So this is what we're studying, to look at all the cells which have been dividing uncontrollably and see what the actual content is of the chromosomes that are inside those cells. So that's what we want to study. And this is what you would get if we would do the same trick again with making a picture of all the chromosomes, so getting a scissor and cutting them out and putting them side by side. Suddenly, it's not just pairs, it's just, well, it's a complete mess. And you can imagine, of course, if you have too much of a specific chromosomes, you have maybe an overexpression of a certain gene and things will get messed up. But also sometimes you have just one copy here, for instance, of chromosome 9, then you are going to miss out on certain functionality of your genome. So this is what you do not want to see, and generally what we find within a cancerous cell. So what do we have at our disposal to actually try and study this? So here at the hospital, not sure if many of you know, we have a pathology department who have been doing this, and I'll explain in a moment, no worries, for years and years and years. So let me just pronounce it: formalin-fixed paraffin embedded material. It's when a patient comes to the hospital and the tumor is resected. What happens to the tissue that's being resected from the patient? It goes to formalin fixation, and after that it's going to be embedded in paraffin. So why? Why do we do that? It is actually routine material for diagnostics of solid tumors. So this is what all pathologists regularly use to do their diagnostics. Because, well, I would put it also there's really a large collection of these paraffin blocks somewhere in the basement of the hospital, and that's really a very, very invaluable resource for us to have. So what actually happens is that this piece of FFPE material is being cut into thin slices, and that's being positioned on top of a microscope spline. At that point, what they can do is they can apply different kinds of staining or coloring, making it feasible for a pathologist to actually study them under a microscope to see what the tissue looks like, what's the morphology of the cells, and on the basis of that, try and figure out if it's a very aggressive tumor, or maybe just a mild tumor, or maybe a different subtype of tumor. What they can then do is actually demarcate the cells which they find interesting, or they find being of tumor origin. And what we can then do is we can actually scrape off these cells which have been demarcated by a pathologist to do further molecular analysis on. So one other very important aspect of paraffin material, the fact that we've been doing that for such a long time, also means that we know what has happened to the patient. I mean, we have blocks maybe from 30 years ago. So we know exactly from those patients, did they survive? Or did they actually, unfortunately, die maybe after the first year? We also know what kind of treatment they have received at that time. So putting all things together, we can actually try and figure out what is the most effective treatment and what is actually the content of the copy number aberrations within those patients. So in that respect, you can imagine that it's a very valuable resource to do research on. But of course, there's also some disadvantages. So if you put tissue in formalin fixation, a number of things happen. The DNA quality is very, 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 very compromised at that point. It causes it to have cross-links, so it really is glued together, and it's very hard to take that apart again. And on top of that, the DNA is not anymore intact, so it's really cut up into millions of little pieces, which makes the analysis, of course, more difficult to perform. So just for instance, if it's a small piece of DNA originating from a highly repetitive region, we have no clue where to put it. But in the end, we can cope. Let's see, because of next generation sequencing. Who has heard of next generation sequencing? Great, not all of you. So I'll just run through this quickly. Actually, the funny part of it is I just mentioned fragmented DNA, which is also the first thing that we perform when we do next generation sequencing. Even if you would have a complete molecule of DNA, we still are cutting it up into little pieces. And that's just to make it, what's the word? Yeah, well, to allow us to actually analyze those little fragments. Because what we have here with next generation sequencing is short read sequencing. We are only able to generate short tags, to generate a short length of DNA, which we take from the molecules. So what happens? So we cut it up into little pieces, and then we ligate universal adapters to both sides of the molecule. And what happens next is we actually throw it on a glass substrate, and it will bind randomly on several positions, allowing us to to sequence really millions, billions of tags at the same time. This actually allows us, if you compare it to our efforts to build the first human genome, to sequence a genome within maybe a couple of days. Compared to 10 years, that's really a very, very big step. So what happens on a glass slide? Of course, we analyze all the molecules which have been hybridized on a glass substrate, and we actually start incorporating nucleotides, and each of those nucleotides have a specific color. So, judging from the specific color and also the specific coordinate of the molecule on the glass substrate, we can start building up the sequence of the reads. So here, for this molecule, at the top left position, we have an AGT, etc. Going to the bottom right position, we have CCT. So that's how it works, but then multiply this by millions and millions and billions and billions. So these are the machines that we're doing it, that we're performing the experiments on. So it's just a collections of specifications, and I think the most important one is the one at the bottom. Nowadays, we were able to generate a complete genome for roughly 2,000 euros. And this is actually the slide that I always need to update each year, because of course there is a new machine already available, which brings down the price to 1,500 euros per genome. And this is just this pricing, and it's not including the actual acquisition of the machine, which is also very, very expensive. So you might have heard about the $1,000 genome. And if you would look at this, you say, oh, we're not quite there yet. But there, you don't actually want to know what it actually costs to build the chemistry and this glass slide to perform the sequencing. It's very, very little. So the $1,000 genome is actually already there. The only thing is that we have to deal with a company which is really very dominant on the market. So they have, like, I think 95, maybe even more percent of the market in their hands, which allows them to actually dictate what the pricing will do for sequencing. Very bad. Evil company. So hopefully, somewhere in the near future, we will have a new technology rising, which hopefully will be able to compete with this company. And actually, we thought we had that with a certain company which is called Pacific Biosciences. Who heard of Pacific Biosciences? Cool. And what happened to Pacific Biosciences?
Illumina bought them.
Uh-huh. So we're back at square one. But let's hope that...
I mentioned another chance.
Another
Hopefully. Nanoperoxide.
Oh, yeah. Another chance for Illumina to just acquire.
Oh, well, probably.
But that's, I think that's a very nice one to mention as well, just on a small side step over here. Because what you need and what makes this machine so expensive are two things. One, the fact that you need to have reagents flowing through this, over this glass slide. So you need a very intricate pumping mechanism, which will never fail, but of course it does not happen. So it gets very expensive. And the other part is that you need a very expensive optics system. So you need to make photographs of all these, of the complete area, while being able to distinguish the different fluorescent dyes, but also the very small clusters that are actually populated on the glass substrate over here. Both of those things are not present in a nanopore system. It just takes in the molecule of DNA, it runs it through a pore, and it directly sequences the content of your molecule. No imaging, no chemistry, and that makes it a very, very, very interesting technology. And we're hoping to get our hands on that. We're hoping that they will develop into quite a contender, but we'll have to wait and see what happens to that company once they get to that level of quality. Okay. So I mentioned we're talking about copy number aberrations, which we will be focusing on in this lecture. I want to emphasize that, of course, it's not the only thing that happens to a genome. Of course, you also have point mutations, smaller aberrations, nucleotides what's the word? Help me out. SNPs? Yeah, but what's the word? Yes, 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 but you know, that you swap in one nucleotide for the other. Mismatch. Oh, yeah. Well, you get the point, I guess. And depending on which type of tumor you're studying, it's more likely to be driven by either mutations, point mutations, small mutations, or larger copy number aberrations. Here you can see that Kirk is kidney or renal cancer on one side of the spectrum, so largely driven by point mutations, whereas on the other side, OV, ovarian cancer, is far more driven by copy number aberrations. So this is just to point out that, of course, we are focusing in on one part of the problem. Do not forget there's a whole other part to it as well, but maybe for some other time. So also to illustrate that, of course, here on the left side, I mean, you have the point mutations. So at one single point in your genome, you see that here is the reference. We were expecting an A over there, but if you would align all the reads that you sequence from the sequencer and put them on top of the reference, you will see that all of them will have a different nucleotide over there. So that's a point mutation. Over here you have small insertions and deletions, which also happen. And maybe on the other side of the spectrum over here, you see that maybe sometimes a chromosome breaks apart and gets glued together, well, not correctly, so to speak. So suddenly it's chromosome 5 attached to chromosome 1, which I think you can imagine can also have some implications on the cell. But what we will be focusing on is, no, it's gone. No, okay. Okay, I had a nice animation over here which would highlight this part over here. And here is just looking at the abundance of the reads that you acquired from the sequencer. You align them to the genome and see how many you have. Maybe at this point suddenly you realize, hey, I have no DNA, I could not find any DNA at this location. Something is wrong, maybe a homozygous deletion. I did find something, but it's not quite as much or as many as I would expect. Then you will have a homozygous deletion, or you find a region where you have far more reads in your hands than you would expect. Again, what would that look like if you, is it broken? Okay. If you would analyze that. So this is the IGV browser. Again, raise your hands who has ever heard of it. Cool.
Never or
ever? Cool. Well, just okay, for the rest of you also. This is what it looks like, and it's actually just a viewer to see what your alignment looks like. Where you have millions of tags, millions of molecules of DNA that you've sequenced, and which the first thing that you do with them, and Jaap just mentioned that in the previous lecture, you use BWA to put it somewhere on the genome. And that's what you see over here. These gray bars are the locations where those reads actually are placed on the genome. Here you see that you get a representation of how much of them are placed at that location. And then just to give you some more context, we are looking at chromosome 18. This is the complete chromosome that you see over here. We are zoomed in on this red bar over here, and we are looking at this sample, which is the tumor. And what we actually do with copy number sequencing is we make predefined non-overlapping bins, and we just simply count what's in there. It's as simple as that, actually. So we take this and we start counting. Not by hand, of course, we have nice scripts for that. And we implemented that in this nice little publication published in Genome Research, where we took that little idea, applied some neat tips and tricks, or neat tricks to make the data more analyzable, and that's what Eric will tell you about in a moment. Just one thing to highlight, we really applied and used tools like bioinformatics and R, and everybody raise your hand because everybody heard of that. Yes? No? Okay. And GitHub to really make it available to the community as well. If any other group at a different institution or different country would like to make sense of what we're doing, they can do that, of course, by looking into the code. But also, if they want to make any addition, improvement, this would also allow them to do so. So I think at this point I'll hand over the word to Eric.
Thank you. Yeah, so I'll describe the QDNAC package that you'll be working with in the tutorial. And this is a nice brief overview of the article, graphical overview. But I'll start first here. So this is data, raw, well, not raw data, but close to raw data from a tumor. And it's very simple. This is already kind of what the OOT showed with the aligned reads. And just simply counting in 15,000 base pair bins how many reads you find, and then taking the log-to ratio and normalizing it. And you can see that it really reads a discrete number here by seeing, oh, this is a couple of reads, this is one more, one extra, and they get closer and close together because it's a log-to ratio. Yeah?
Why did you use non-overlapping regions and not like a window, a sliding window?
It's a good question. I think it's just because it was simply simple to implement, and also the other thing is the GC correction will probably be a bit easier. But a sliding window approach would probably work as well. I think at some point there were sliding windows in QDNAC.
Yeah. So I think this is very much a practical consideration, because if you have sliding windows, the resolution or the number of data points you will get can go up to each nucleotide, of course. And this is just to make it more feasible for us to perform. And also, if you would follow the story of Eric, you will see that doing it in this way already gives you a very good resolution compared to the method that we used previously.
Another thing to highlight, I will highlight it later, but it makes the quality control a bit easier as well, I think. Because every bin is now independent from each other. But even just simply counting, so not doing anything else, you can already see that something is happening on chromosome 10. There are less reads than you'd expect. And you see some other regions where maybe something is going on. And there are also quite a few reads where there are a lot less than you might expect. So, but you also see that it's quite noisy. It's noisy than, more noisy than profiles that you will see later on. So the first question is, where does the noise come from? Now, one is the genome is randomly fragmented, so there's just a bunch of places, yeah, there's just random variation, you can't really do much about that. But there's also systematic noise, and two of the most important ones are GC content and mappability, and I'll show you how we'll deal with that in the next couple of slides, one by one. So the first is the GC content. So there are 4 bases, as you know, and these two bases make a different kind of, have less hydrogen bonds, so they are less stable than the GC bond, the cytosine guanine. And the fact that there are 3 bases here is as a consequence. They have a higher melting point. They break, so these break apart quicker, and because of that, you get more reads than you would expect from just randomly fragmenting the genome with GC bias. And the other thing is that this degrades slower. And as we're working with FFPE material, we're working with degraded DNA. So there's also bias caused by that. The other one is mappability. So let's say you have a read, and you try to map it to the genome. But as it's a short read, there could be a homolog or some other repetitive region that goes into 2 places. So you have this part in chromosome 1 where it aligns, and this one in chromosome 12. Now, where did it come from? There's just no way to tell. So how do you deal with this? Well, it's in fact very easy, because we have the reference genome. We can just calculate the GC content and the mappability for each position in the chromosome, and then see how many reads you'd expect based on those parameters. Are they less or more than you'd expect just from random variation? And if you know how many more there are, you just multiply the signal by that number, and you improve the signaling. So this is before correction, and this is after correction. and you see that it improves slightly, but there are still a lot of regions that that don't work out well. So then we started with the blacklist, basically removing bins from the analysis that that we couldn't sequence well, and of course you need to have a certain amount of so you need to have reasons to remove a bin. So the first step is removing bins with mappability below 50. So that means that on average each read in that bin fits into another bin as well. So at least 2 positions that a read maps to. And that is incorporating mismatches from the system. So by this you get rid of repetitive regions, for example. And the other one is simply looking at 1,000 healthy genomes, or actually we took 38 because that's quicker to calculate, and we needed similar chemistry and in our analysis as we are using, and see if there are bins that are higher than you'd expect, because the healthy subjects don't have any copy number changes. So those are removed, and then we went to a public blacklist that included a lot of hypervariable regions. So they just differ from person to person, and that means that the reference genome, they have to take some consensus, but if there is a region that's a lot longer for an individual, you won't be able to tell if there were more reads than expected or if the region is just longer. So those are blacklisted as well. So these are the bins that are blacklisted for the different reasons, and you can see that they tend to be the ones that are outside of normal places, away from 0 essentially. So if you do all that together, then you essentially get a nice CSI moment. So this is the original profile, and now we do all these steps, and we enhance the image. So now you can see a lot more clear where homozygous deletions are in EGFR amplification, which is very common in cancers. So this is log2 ratio, remember, so this gene actually has 32 copies somehow. And also a gene with 2 losses. So this is log2 ratio, so why do you think this is actually not at minus infinite if you have a ratio count? Can anyone guess? Yeah.
There will also be some normal cells on the side.
Exactly. Yeah.
Can you repeat what you said?
Oh, there are some normal cells in mixed in with the tumor, so that dilutes the signal. And in fact, that's an issue. Some tumors are very spread out over normal, so we can't see anything at all because of this. So it's also sometimes a big problem. Okay, so now that we have this, you have a copy number profile, and of course the world is not perfect. You also have always have some noisy profiles. And so we already improved this by GC content, correcting for GC bias and mappability. But sometimes you still get noise. And with this we distinguish between statistical noise, so noise that comes from reads being placed in different places randomly, and a bias. So some regions get more reads than you'd expect by chance than other regions. And the way we distinguish from this is by looking at these 2 numbers. I'll go into a bit more detail on that later, but basically the E stands for the expected noise from by chance, and the sigma stands for the noise we actually observed. And this is actually what we want to see. So a low noise sample. So yeah. Okay, so first we need a measure of how big the noise is, and normally you take something like the standard deviation of the bins. But as you can see from, for example, this is a high quality profile. Yeah, sometimes you just have changes in the profile that are due to biological origin, so that doesn't work. So the algorithm that's used is look at the differences between 2 consecutive bins. So for example, between this bin and this bin, between 2 dots, and calculate the average of those, and then remove 0.1% of the most extreme values, because those are likely the real copy number changes. Now calculate the standard deviation of the differences, and then there's a mathematical formula for the expected variance, which is simply the number of bins divided by the number of reads. So if you have more reads, you have a lower expected variance. Now, and we do this, we compare the observed variance, so the ones we calculate for the real profiles, and with the expected variance, and that's a measure of the DNA quality. So just to summarize, what you should do if you have done this quality assessment. So if there's a low observed and a high expected noise, that's something that goes against the laws of statistics. That means you basically get exactly the number of reads that you'd expect, but there's no variation that you'd expect. It's essentially if you throw a dice and you get 1, 2, 3, 4, 5, 6, exactly the same amounts of times. You expect some variation. So this never happens. The ideal situation, low observed noise and low expected noise. A high observed and high expected noise, that means you have few reads. So that means that you just put the sample on a sequencer again, if you can. And then a high observed and a low expected noise, there's not really a solution here. It basically means that there's an intrinsic bias in the data that we don't know where it's from because we already corrected for GC and mapability, the two that we know. Right, so, okay. So these are representative examples. So low observed, low accepted noise, high observed, high expected, and systematic bias. So these are all samples from real data, and most of the samples you see that they're at this line of observed and expected noise. Sorry, expected and observed noise. So ideally they're on this line, and there are always some outliers with probably degraded DNA or some other reason that there's more noise than you would expect. I will actually skip this, I think, because I'm running out of time a little bit. So this is really briefly, this is the wave step to get rid of the to get rid of the systematic bias. We sequenced a bunch of healthies and checked if we found some systematic bias in there, and based on that corrected tumor profiles. So that's these profiles, and you can see from here essentially again a Lowe's correction based on the same parameters. So these are healthies, and there's still some pattern in there, and we think it's due to, well, we don't know. So there have been some recent results that put the biological origin of this into question, but the idea here is that some parts of the genome replicate earlier. So if you some parts of the genome systematically have a higher number of copies than other parts, and this synchronizes, and that's why you see the systematic bias. But there's no way to really tell this. So in practice, we just observe some noise and subtract it from the copy number profiles. So if you do that, you actually see that you can improve the profile again, just like with the GC content correction and the mapability correction. The only difference is that now we don't know what we're actually correcting, only that it's been in the profiles. This is actually not in the practical, because this is something that differs from tumor type to tumor type, for example, and tissue type to tissue type. And we don't actually know where it comes from. Then the follow-up processing. Segmentation. So now you have all these different profiles, and where are the breakpoints of the genome. So this is done not by qDNA-seq, but by DNA copy, which was already published, but it's incorporated in qDNA-seq. And calling. So now that you have these lines here, you know where the breakpoints are. What is a gain? An extra copy. What is an amplification? Multiple extra copies. No change. A loss. Lose one copy. A deletion. Lose two copies. So this is what it looks like. These are gains, and these are losses, or at least as called by the algorithm. I should note that especially this step, there are a lot of competing algorithms. The first two steps are more or less accepted, how to do it, and most algorithms do it the same way, but here, going from a copy number profile to a discrete number of copies in chromosomes, there are a lot of different methods out there that do this. And we also see that from other people that adopt QDNASI, they tend to swap out this part from QDNASI. So there is a lot of literature on how to do that correctly, and some of it also uses mutation data to actually find out how many copies there are. And the difficult thing is that some tumors are actually tetraploid instead of diploid. So that makes it a lot more complicated. And there's also heterozygosity in tumors. So sometimes half the tumor has a gain and the other half is neutral. And it's really hard to find the difference or tell the difference between infiltration from normals, extra copies, so tetraploidy, and heterogeneity in the tumor. So half the tumor has this. It's almost impossible to do all of this simultaneously. And that's the reason there are lots of different methods, and this is an active area of research, I think. So, the QDNASI practical, there are a bunch of cancer cell lines, and you can actually go from raw sequencing data to copy number profiles. And raw sequencing data can either be FASTQ files, which are the reads, or BOM files, which are the aligned reads. Depending on where you get your sequencing data from, you get either FASTQ files or BOM files, and you can always go back from BOM files to FASTQ files. I don't know if you actually do the alignment step in the practical. I think it depends from year to year. And that's actually not part of QDNASI. So QDNASI starts from BOM files, aligned reads. QDNASI is already installed on practical machines, but for those interested, you can obtain it freely from Bioconductor, including a reference manual and a tutorial. And the source code is available here. And I think if you do become a researcher, and I think a lot of you actually will become, it's actually a nice to learn about Git and Bioconductor because almost all open source projects nowadays use GitHub to publish their data, and also if you want to make a contribution, you need to do it through Git. So I'd like to encourage you to put up your tools online and learn how to work with it, because it is sometimes a bit difficult to work with it. So very quickly, I think I'm already over time a little bit. Yeah. So if you do put your tools that you developed in your group online, what can happen is that people actually adapt your tool and use it to do really cool research. So this is not from our group. This guy actually moved to our group before he worked on this, and it was on the cover of Science Translational Medicine last week. Research that we actually, as a group, didn't know about, and they used QDNASI, or at least a part of QDNASI. And what they did is they developed a method to obtain copy number profiles from blood samples of cancer patients. And that's actually harder to do because, as you can imagine, the DNA is a lot more diluted than in normal from a tumor, if you take a biopsy from a tumor. And to do this, they started with xenograft mice, which are mice with human tumors implanted in them. And they tried to see if they could find differences in the DNA that map to the mouse, and the reads that map to the mouse genome, and the reads that map to the human genome. And the nice thing here is that you know that the human reads are definitely for tumors, from growing cells. So, and what you can see is that the reads that map to the human genome are actually a lot shorter than the reads that map to the mouse genome. And what you can also see is that the fragments are a lot shorter than we're used to. They're only about 150 base pairs. So they implemented the size selection to do that you need parent sequencing to know the length of the read. And you can see that without size selection, you see some changes maybe, but they're very very flat and very hard to distinguish from noise. And if you do these size selections where you only take into account short reads, then you suddenly see all these amplification, gains and losses. And you can also see that it's very heterogeneous. You have a lot of different levels. So, take-home messages. Whole genome sequencing is very cost effective. Actually, we've done now 100 genomes because of the low coverage for €100 a piece. And that's because we only get the copy number, so you need less coverage. We can use archival FFP material with this method. QDNA-seq starts from raw output, depending on what you call raw. It starts from BOM files, which most sequencing facilities actually deliver to you. And yeah, the noise estimate, so what makes QDNA-seq unique? Noise estimate, 2D mappability GC correction, most of the methods do it sequentially, and it's modular, available, and open source. And just acknowledging everyone, so I'm part of the tumor genome analysis core, so the part of the pathology. My supervisor, Bauke Elstra, was involved with this project from the start. Florent, the guy that was on the cover, actually moved to our department, also recently. And the clinical genetics, which are a lot closer to the diagnostics. Those, of course, that also worked on this project. So, thank you, and I hope you have fun with practical.
30 seconds. So if you like this stuff, you're going to work with real live data, right? For the first time. So it might be so. Now it is the coordinator of bioinformatics research, clinical genetics. So if you like this stuff a lot, maybe if you want to do an internship, talk to the man. Not guaranteeing he has time to try. This is a point-in case as it comes to inspiration. This gentleman was, I think, a master student bioinformatics in our department. Then he went on doing a PhD in our department. And after that, he got stuck at the VMC. That's a fantastic place to be. So there is hope, guys.